Today, our topic of discussion is urea cycle. Rather, we can say that it is amino acid catabolism. So, amino acid in general do not undergo a catabolism process. Catabolism of amino acids takes place only under certain conditions. When we are taking, suppose, surplus amount of protein, or we are taking a keto diet or a protein high protein diet, then only the catabolism of the surplus amount of amino acids or the proteins which we take is going to occur. Simultaneously, when a patient is suffering from diabetes mellitus or type T2DM, which is type 2 diabetes mellitus, there is carbohydrate which cannot be absorbed by the body, and therefore the body starts using up the reserve amount of proteins, which are the protein, suppose the muscle cells or the other reserve proteins, and they start the catabolism of those and to get energy from the protein molecules. So these are the conditions, these are special pathological conditions where there is this T2DM or type 2 diabetes mellitus is a condition, is a pathological condition where there is catabolism of protein taking place. Now, there are balances in the protein, like it is known as the nitrogen balance. So, nitrogen balance is basically what we check for seeing the concentration of protein, or rather for checking the protein catabolism rate. In a healthy individual, generally nitrogen input is equal to nitrogen output. Rather, protein intake is equal to protein that leaves the body by the process of after the process of catabolism, which is getting excreted. So nitrogen input is equal to nitrogen output in the case of a healthy individual is nitrogen input is greater than nitrogen output in the case of a growing child. In the case of growing child who is requiring high amount of protein, or a person who is recovering after any massive surgery or something, requires more of protein diet, therefore the nitrogen input is greater than the nitrogen output. And in certain conditions, pathological conditions, nitrogen output is more than the nitrogen input. Therefore, huge amount of protein is getting excreted out. This may be due to some metabolic error in the body. So this is what, because this is the nitrogen balance which we actually check for the seeing the level of catabolism which is taking place inside the body. Now, as we have already discussed, the reactions of proteins like transamination, oxidative deamination, non-oxidative deamination, transmethylation, all these reactions primarily leads to formation of a product which is ammonia. But ammonia, as we all know, is basically toxic to the cell, and we humans are basically ammonotelic, because we are not ammonotelic, sorry, we are ureotelic, because ammonia we cannot excrete in the form, we cannot excrete the waste product of protein metabolism in the form of ammonia. So we are basically ureotelic, therefore we have to convert the ammonia which is present inside the body, or which is being formed after the catabolism of the proteins, needs to be excreted from the body. Before excretion, it needs to be converted to urea. So today our primary focus is going to be the urea cycle, which is the one which is taking place, and which is the most important cycle for getting rid of the ammonia. But before that, we have to discuss certain things, like the ammonia basically is a very toxic substance which is present inside our body. So ammonia as such cannot be present inside the body in a high concentration. Therefore, ammonia, before it undergoes the urea cycle, first the ammonia has to form something called glutamine, because glutamine is a form where there is ammonia which is a coupled form of ammonia with glutamate, which actually will help in the elimination, or rather it is a better form which is a non-toxic form, because as we have already discussed, ammonia is toxic and therefore it has got some adverse effect inside the body. So like if ammonia increases inside the body, it acts as an osmotically active solute, and therefore it will draw more amount of water inside the astrocyte, or it's a kind of brain cells, and therefore it leads to the effusion in the brain, or it can lead to water accumulation in the brain, which is harmful for the brain and can lead to the comatose stage. So we are not actually in a position to let ammonia accommodate inside the body, and therefore we have to convert the ammonia immediately into some form which is a non-toxic form. Therefore ammonia, which is basically the form which is actually going to be transported into glutamate. So glutamate, rather L-glutamate, by the action of glutamine synthetase. First, here there will be conversion of ATP to ADP to form gamma glutamyl phosphate. To gamma glutamyl phosphate, again glutamine synthetase acts, and now there will be entry of NH4 plus molecule, PIB will get released, and therefore leading to the formation of glutamine. This glutamine is a form which is actually a form which is going to be an harmless, so it is a non-toxic form. It is a non-toxic form and it is not going to affect. So first we need to convert glutamine, with the help of glutamate we need to convert the ammonia, which is a toxic form, into glutamine, which is a non-toxic form. So this is a process which has to take place. Now before moving further, now the most important thing, that this ammonia which is being produced by most of the reactions of amino acid catabolism cannot be used by all the reactions. It can only be used by 3 of the reactions which are catabolized by 3 specific, which are getting catalyzed by specific enzymes. So fates of ammonia. Ammonia can be used up by 3 reactions. One is a reaction which is catalyzed by the glutamate dehydrogenase. As we have discussed previous day about the glutamate dehydrogenase reaction, which is actually an oxidative deamination reaction, but today we are going to discuss glutamate dehydrogenase, but it's opposite to the oxidative deamination reaction, and so it is known as the reductive amination reaction. It is just the reverse. So reverse of the glutamate dehydrogenase reaction which we have done the previous day. Here actually alpha ketoglutarate will react with NH4 plus to form glutamate. This is a basic reaction. Here there is NADPH which is getting converted to NADP. So this is a basic reaction of the glutamate dehydrogenase. It is a reductive amination reaction. So this is a reductive amination reaction. Now the next reaction which can use is the glutamine synthetase, which we have just discussed previously. And the third reaction which we will discuss in greater details, so I am not going to write into much of details in this place, is the reaction which is catalyzed by the carbamoyl phosphate synthetase. Carbamoyl phosphate synthetase is an enzyme which is the first enzyme of the urea cycle, which is basically required for the transfer, or rather being for the formation of carbamoyl phosphate from the molecule of urea. So these are basically the 3 reactions which are required for the ammonia to be disposed, or these are the reactions which actually can dispose the ammonia. For a brain cell which is entering the comatose stage, where there is excess accumulation of ammonia, the first reaction that is going to take place is going to be the reductive amination reaction, where there will be the formation of the glutamate, so that the glutamate can be, with the help of the glutamine synthetase reaction, can form glutamine. So now we can move towards the proper urea cycle. Urea cycle is the main process where we actually where the protein molecule is disposed. So this process basically is taking place inside the mitochondria and cytosol. So we can rather say that this is the compartmentalized reaction. So the urea cycle is going to take place both in the mitochondria and also in the partly in the mitochondria, partly in the cytoplasm. There is the first main component, first main enzyme which is required for the urea cycle is basically carbamoyl phosphate. In the case of eukaryotes, we see that there are basically two types of carbamoyl phosphate. Carbamoyl phosphate 1 carbamoyl phosphate 1 carbamoyl phosphate 2. There is a difference between the carbamoyl phosphate 1 and carbamoyl phosphate 2. Carbamoyl phosphate 1 is the one which is leading to the formation of the carbamoyl phosphate. Carbamoyl phosphate 2 is getting utilized for the synthesis of pyrimidine, or rather it's involved in nucleotide biosynthesis, involved in the nucleotide biosynthesis pathway. And this is with the urea biosynthesis, the urea cycle. It is involved in the urea cycle. This is the nucleotide biosynthesis. This is using NH3 as a source of nitrogen, and this is using glutamine as a source directly. So there is a difference between the two, carbamoyl phosphate synthetase 1 and carbamoyl phosphate synthetase 2, both required for various different functions. So now, what are the reactions? What is the reaction which is getting catalyzed by carbamoyl phosphate synthetase? Because we are not concerned with carbamoyl phosphate synthetase 2 at all in this reactions. So what is carbamoyl phosphate synthetase? First, what are the reactions? The first, we are going to concentrate on the reactions which are going to take place inside the mitochondria. So what happens is, firstly, we are going to dispose of two molecules of ammonia and two molecules of bicarbonate, or rather carbon dioxide in the form of bicarbonate ions. So first reaction is going to be this is known as carboxyphosphate. Now there is an entry of an NH3 molecule, and it will lead to, and there will be elimination of a Pi molecule, leading to formation of, so this is what is known as the carbamate. Carbamate now is again going to react with another molecule of ATP. The reaction is being catabolized by the same enzyme, which is carbamoyl phosphate synthetase, to form a product So this is what we are going to, this is what we are concerned, and this is what is known as the carbamoyl phosphate. So here first a molecule of ATP reacts with bicarbonate, rather it's HCO3 H2CO3, and then it finally reacts with carbamoyl phosphate synthetase to form carboxyphosphate. Carboxyphosphate, there is an elimination of a phosphate group, there is formation of carbamate. Carbamate then forms carbamoyl phosphate. this carbamyl phosphate is going to be utilized in the subsequent reactions of the urea cycle. So now this is the most important part. So this is the first step of the urea cycle, which is taking place inside the mitochondria. Now this carbamyl phosphate which is getting formed will now react with this is known as ornithine. This carbamyl phosphate reacts with ornithine in the presence of an enzyme known as ornithine transcarbamylase. Ornithine is a protein molecule, an amino acid which is a non-standard amino acid because it is not among the 20 primary amino acids. It is only present inside the, or rather it's only formed in the case where there is in the urea cycle. So it is an intermediate of urea cycle. So this ornithine is basically a product which is important for the formation of arginine, in the breakdown of arginine, in the formation of urea, and therefore it's a precursor of the urea cycle. So this carbamyl phosphate reacts with ornithine in the presence of ornithine transcarbamylase. This group, which is the CONH group, is known as the carbamyl group. So this carbamyl group is going to get transferred over here, therefore leading to a compound which is this is what it will look like. This is what is known as the citrulline. So ornithine transcarbamylase reacts with ornithine transcarbamylase is an enzyme which basically catalyzes carbamyl phosphate with ornithine to form citrulline. This citrulline is what formed inside the mitochondria will be transported out of the mitochondria with the help of certain transporters to the cytosol. So it is excreted to the cytosol. And the next reactions of the urea cycle will take place inside the cytosol. Now this citrulline which is being formed in the mitochondria and excreted to the cytosol now will react with a molecule of ATP. So this molecule of ATP which goes and attacks at this carbon, and therefore this can be rather better to write it as CONH2, and it will attack at that carbon and lead to formation of COAMP NH2 NH. Here there will be a double bond which is present, and NH, and then there will be CH2 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 CH NH3 COO minus. This is what is known as the citrulline AMP intermediate. So after citrulline is it's worked upon by ATP with the help of enzyme arginino succinate synthase, an enzyme called arginino succinate Synthase is an enzyme which is basically catalyzing citrulline ATP reaction to form citrulline AMP intermediate, which is then acted upon by an enzyme. Again, with the same enzyme, here we have one molecule of aspartate, which is nothing but COOCH2 CHNH3 COO minus. Now in this reaction, this will go and attack here, and we will have a release of one molecule of AMP from here, and now it will form nothing but this is what is known as the argininosuccinate. This is a compound which is known as the argininosuccinate. It's leading to the formation of argininosuccinate. So what is happening first, this is the second step, rather it is the third step of the urea cycle, where citrulline acted upon by ATP forms citrulline AMP intermediate, which again reacts with a molecule of aspartate to form argininosuccinate, which is an intermediate again, with the help of enzyme argininosuccinate synthase. Now argininosuccinate which is formed will be reacted upon with the help of enzyme argininosuccinase, and it will lead to the formation of this is known as arginine, and with it we get fumarate. So we get fumarate along with this. So what happens basically is, here there is a reaction with argininosuccinase where we get arginine, and we also get fumarate, which this fumarate which is formed will enter, then which will subsequently form malate and therefore enter the gluconeogenesis pathway. Therefore this fumarate goes into the gluconeogenesis pathway. So argininosuccinate with the help of enzyme argininosuccinase leads to the formation of arginine and gluconeogenesis, and fumarate which will ultimately form malate and then enter the gluconeogenesis pathway. So this is basically the reactions of now last step is this arginine will be acted upon by an enzyme called arginase, and this is the last step where arginine is going to be acted upon by an enzyme called arginase to form one which is nothing but ornithine again, along with it is going to form which is nothing but urea. So finally it is leading to the formation of a product which is urea, which is NH2CO NH2. So ornithine is being formed. Along with it also we are getting the formation of urea, which can then be excreted out of the bodies, which can then be excreted out of the body. So this compartmentalization means the first two reactions taking place inside the mitochondria, and the next three or the last three reactions are basically taking place inside the cytosol. So the enzymes which are required, firstly we have carbamyl phosphate synthetase 1. Next we have ornithine transcarbamylase for the conversion of ornithine plus carbamyl phosphate to form citrulline. Then we have argininosuccinate synthetase for the conversion of citrulline, rather from citrulline AMP intermediate, and finally to the formation of argininosuccinate. Then we have the action of an enzyme called argininosuccinase, which will lead to the formation of arginine, and then we have an enzyme called arginase, which will lead to the breakdown of arginine to form urea and also ornithine. So this completes the urea cycle. So here basically we use two molecules of ATP molecules which are getting utilized in the first step, in the step catalyzed by carbamyl phosphate synthetase 1. So that is the step where there is two molecules of ATP molecules are getting utilized. Now coming to the control, this urea cycle requires a compound called N-acetyl glutamate to activate the carbamyl phosphate synthetase. So N-acetyl glutamate, which is formed by an enzyme N-acetyl glutamate synthetase with the help of reaction catalyzed by acetyl CoA plus glutamate, reacts with the enzyme acetyl glutamate synthetase to form N-acetyl glutamate. So this reaction is required, and this N-acetyl glutamate is an allosteric activator of the enzyme carbamyl phosphate synthetase 1. So N-acetyl glutamate is very much required for the urea cycle to take place. Without the presence of N-acetyl glutamate, we cannot proceed further with the reactions. Now there are certain diseases which are accompanied by the urea cycle, which are due to the defects of the urea cycle, like it can be citrullinemia, it can be ammonemia or hyperammonia 1, type 1 hyperammonemia, can be type 2 hyperammonemia, and it can be arginemia and etc. So there are the conditions where there are the defects of several enzymes associated with the urea cycle. Now, if there is a defect in the urea cycle enzymes, there are certain treatments which can be given. Suppose we can give hipurate. Hipurate can be given to a person with urea cycle enzyme deficiency. Moreover, a person who is deficient in the urea cycle can also be supplemented with arginine. Arginine supplementation is also very important, or it's very useful to patients who are suffering from urea cycle enzyme deficiency or a metabolic disorder of urea cycle metabolism. So these are the two compounds which can be used. So now lastly what we have is that the compartmentalization, as we have already discussed, that this carbamyl phosphate which is basically carbamyl phosphate synthetase 1 and 2 which are there, carbamyl phosphate 1 which is required for the synthesis of urea cycle, or rather of urea, whereas the carbamyl phosphate 2 is involved in the synthesis of pyrimidine nucleotide in the DNA RNA metabolism pathway. And lastly, the fumarate which is being formed in the urea cycle basically enters the rather the gluconeogenesis pathway, or rather that can also enter the TCA cycle. So it is known as argininosuccinate shunt of TCA cycle. It is known as the argininosuccinate shunt pathway. So by this we can conclude our session for the day, because this is it about the urea cycle and the protein metabolism part. So please do subscribe the channel, and thank you.